Hi everyone, this is your instructor Kwame, and this part of the lecture on virals, introduction, and DNA viruses. We'll look at the second part of the intro to viruses, and then we will then look at some viruses that infect humans. So we'll look at the lytic cycle. So in this case, we're looking at viruses that infect bacteria. The lytic cycle has these steps in terms of the cycle of the virus. There's an attachment phase where the cell surface receptors of the bacteria would come into contact with the receptors on the surface of the virus. Note that the virus doesn't play any active role in seeking the host. It is just a chance encounter. Then there is penetration, where only the DNA or nucleic acid would enter. The capsid would enter as well, but the capsid would then be degraded once it enters. The component of the virus that is essential for its replication and survival is the genome, where it has its DNA or RNA sequences. Then there is biosynthesis, where there is a production of what we call a phage DNA. The phage DNA, it's the point where you have the virus DNA incorporated into the host DNA. And then you have the proteins as well. There is maturation, where the components of the virus are then assembled, and then there is a release. So I look at it this way. There's a locate and stick. Then it has to get in and then make pieces of itself, put it together, and then get out of the host. So the penetration is done through enzyme activities, and the release is also done through enzyme activities. So enzymatic activities govern these processes. So to expand on the steps again, it will come down to what type of host cell that the virus is attacking. And in this case, it's a prokaryote. If it's a prokaryote, there's a difference in how it happens. If it's happening in a eukaryote, it's going to be different as well. If it's a prokaryote, you have a phage. If it's a eukaryote, you would have animal or plant viruses. Okay. Then you have the capsid morphology, that will also influence how the attachment and penetration proceeds. There are three different ways that this will happen. For viruses that are naked viruses, they will penetrate the host cell by direct penetration, because there are no barriers. It is just the capsid. There is no envelope. For those that are enveloped, they may use one of either two procedures. Either they will become enveloped. Because they're enveloped, they will become integrated into the membrane of the host cell. And then once they become integrated in the membrane of the host cell, they release their genome into the host cell. So that is known as membrane fusion. And then there is endocytosis. It's more like a form of phagocytosis. But in this case, there is another membrane that is formed using the host cell membrane. It is then formed around the virus envelope, and then it is then brought into the cell. Once it gets into the cell, there is one coating of that the viral membrane, and then degrading of the capsid, and then you have the genome. Pictorially, this is how it presents. So you have a naked virus. There is no envelope around it. So the capsid will come into direct contact with the cell membrane or the cell surface receptors, and it has to be a good fit for any form of penetration to occur. So if it's a good fit, there is attachment, and then once attachment happens, the nucleic acid would then be released into the cell. So this is a direct penetration. Then you have the membrane fusion for enveloped viruses, and they would fuse when they come into contact with the right receptors, refuse with the membrane, become part of the membrane of the host cell, and then the nucleic acid is then released as the capsid is degraded. Then you would have the endocytosis, where the cell membrane or the surface membrane of the host cell engulfs the entire enveloped virus. So this is the enveloped virus. It finds the right receptors and attaches. It is then enveloped. So you have two covering around it. If you look carefully, you can see that. And the two coverings will all be taken into the cell, endocytosis, and then they will then be degraded. The capsid will be degraded, and then you have access to the viral nuclei. So after the entry, the attachment, the penetration, there's then synthesis, there's assembling of it. For that to happen, the bacterial system has to be overcome. So there are some enzymes that have to be synthesized. The entry enzymes, so there are lysozymes that are released. There are polymerases that are also released or synthesized. Those are early enzymes that the virus has to synthesize once it enters the cell. It will then use the polymerase to make copies of itself. It will then be able to synthesize the protein that it needs to continue with its assembly. Once the early proteins have been synthesized and the genetic material is available, which is the DNA or the RNA of the virus, it will then go ahead and create the protein that it needs and then use the genetic material that it has as a template to continue to synthesize the other proteins that it needs. Then all this process, as it's going on, replication is going on, so more copies of the DNA is being made. It will then also come down to the type of cell. The type of cell will influence how this process occurs. If it's a eukaryotic cell, the transcription and assembly, some component of it would take place in the transcription and assembly of the messenger RNA will take place in the nucleus, and then the translation or the processing of the proteins itself will take place in the cytoplasm. Okay. For prokaryotes, everything will take place in the cytoplasm. So pictorially, if we look at a phage, bacteriophage, so if a phage, a virus that is infecting a bacteria or attacking a bacteria, we call these the T-even bacteriophage. It's a very loose name that encompasses, among other strains and isolates, the enterobacteria phage T2, enterobacteria phage T2, enterobacteria phage T4, enterobacteria phage T6. So we call them T-even bacteriophage. So that's just by the way anyway. Now you have a bacteriophage that is attached to a bacteria, and then it will then introduce its nucleic acid, penetrate it, and release its nucleic acid into the host cell. For that to happen, there are a number of things that has to take place within the bacteriophage. This is the component that is important because this is where the nucleic acid is. However, for that to get into the cell, you will need the tail. So this is the tail. The tail is made up of the sheets. These are capsids that you see around it. You have the sheets, and then these are the fibers. There is a base plate over here, and then there is a pin. Now all of them have to work perfectly. Inside the sheet is what we call the core, the tail core. So once it attaches, what will happen is that the sheet will contract. So if you look at it, the length, there is a reduction. It will then contract, and as it contracts, it will then be releasing the genetic material into the cell. Once the genetic material enters the cell, it will then start directing the synthesis of the early proteins and then serve as a template to make more copies of itself, as you can see here. So you have more copies of itself. It's making copies of its capsid as well, and then making copies of the sheets that will constitute the tail. Okay. And all those processes, once they are all ready, then it reaches the stage of maturation. So all the virus components would then be assembled, as you can see over here. So they are put together and assembled, and then it is ready for release. Then it's released, the final stage where enzymes are then used, the cell wall components of it will be lysed, and then the virus will then leave the cell. So that's the lytic cycle. That's the lytic cycle. The lytic cycle is also known as the virulent phase because it causes the death of the cell. And so it will lyse and cause the death of the host cell. So you also have the lysogenic cycle. The lysogenic cycle is also known as the temperate phase. So the lysogenic is also known as the temperate. The lytic is also known as the virulent phage. Now, when the phage DNA is incorporated into the host DNA in the lysogenic cycle, you have what we call the prophage. In the lysogenic cycle, you may have what we call a phage conversion, where the presence of the phage virus would confer on the host cell some characteristics that it hitherto didn't have. So for instance, when we look at streptococcus, the strains that cause septic shock are the strains that have the prophage gene in them. And so that's phage conversion. You could have also a generalized transduction or a specialized transduction. For a generalized transduction, the virus, as it's assembling its components, will pick up components of the bacteria, which it will carry with it, and then when it's infected new bacteria, it will transfer those genetic components to the new bacteria. Then you have the specialized transduction, where as the virus it's excising itself, taking out its genetic components from the bacteria, it takes extra components of the genes of the bacteria, incorporates it into its DNA, and then it's then covered in its capsid. So that's a specialized transduction. The cycle, in terms of the lytic and the lysogenic cycle, pictorially, this is how it happens. So the phage will attach and inject its DNA into the cell. Since this is the lytic cycle, it's going to remain separate from the chromosome of the bacteria. And then the virus will then take over the machinery of the cell, make copies of its own DNA, make its own proteins, and all that, and be able to assemble itself to the point where it will lyse the whole cell and then release itself, and then the cycle continues. And then you have the lysogenic cycle, which is a bit different. So what happens with the lysogenic is that the virus DNA, once it's injected into the cell, will incorporate itself into the chromosome of the bacteria, resulting in what we call the prophage. And then once you have the prophage, Since it's part of the chromosome of the bacteria, when the bacteria is dividing, it will divide with it. So then more copies will make. So there's one cycle. There are instances, however, where through some recombinant events, you could have the virus DNA cut off or excised from the bacterial DNA. But once this stage happens, it will then enter the lytic cycle because the two are separate now. And then the lytic cycle would take over. So in terms of the specialized transduction, it gets to a point where the virus would have to excise its DNA, and so it may be there latent for a very long time, and then something will activate it and it would try to excise its DNA from there, and then proceed to do the lytic cycle. Now, for that to happen, what happens sometimes in terms of the specialized transduction is that it will cut off a bit of the DNA of the chromosome of the host cell and incorporate it into its DNA. And then this would then be covered with its capsid. So it's taking more than it actually brought in terms of the length of its DNA component. So this is specialized transduction, and it confers special property on these viruses so that when they infect a new bacteria, they may have different effects on them as well. This is our participation question for the week. Streptococcus pyogenes, a bacterium, produces an exotoxin that causes scarlet fever only when it is lysogenic. Based on your understanding of the viral life cycle and the results of lysogenic cycle, briefly explain what it means to be lysogenic and how lysogeny allows Streptococcus pyogenes to cause scarlet fever. For envelope viruses, which are mainly animal viruses, when they are exiting the cell, they may do it by the process of budding. And for budding, what it results in is that components of the cell wall of the host is taken together, so it becomes the envelope, and then it would then detach from the cell. So what you end up having, you have the nucleic acid, you have the capsid, and then you have another covering which it takes from the host cell. That's the envelope. For multiplication of DNA to occur, these are the steps that has to happen. So using papovirus as an example, you have the DNA. So there's the DNA. It's surrounded by its capsid. Once it attaches to a host, the genetic material and the capsid will be able to enter the cell. So this is a naked virus. Okay. Enter the cell. Once it enters the cell, the capsid will disintegrate, and then the viral DNA will then take over the machinery of the cell, make messenger RNAs that would encode for early viral proteins, and then use the viral proteins that had been synthesized to make copies of itself. Okay. Now, once it's made copies of itself, it will continue to make copies of the other proteins that it needs to assemble itself, like the capsid proteins. Once everything is ready, it will then assemble itself. So there's assembly, and once it's assembled, it will then be released from the cell. Okay, so that's the cycle. So we'll briefly then look at some DNA viruses that infect humans. So the first group will be the double-stranded DNA viruses. And those viruses may be enveloped or non-enveloped. So the non-enveloped double-stranded DNA viruses will have the family Adenoviridae, adenoviridae because they were first extracted from adenoids. Adenoids are tissues that you find towards the back of the mouth, and they work together with tonsils as part of the defense, part of the immune system of the cell, of the body, sorry. And one important genera that you find in this group is the mastadenovirus. So mastadenovirus. The sizes will range from 70 to 90 nanometer clinically, and what the importance that they have to us as humans is that they cause respiratory infections in humans. Human adenovirus 14, which will be a subtype or a strain, is associated with severe and even fatal outbreaks of pneumonia in residential settings and military bases. Then you have the non-enveloped family virus. Another one would be the Papoviridae. For the important genera within the Papoviridae, we have papilloma virus and polyoma virus. We'll concentrate on the papilloma virus because of the implications that it has on us as humans. Note the size as well, very, very small, 40 to 57 nanometer. Clinically, human papilloma virus is an important pathogen, human pathogen. There are over 100 different strains, but about 30 of these are sexually transmitted. They cause infections in the genital area, including the skin of the penis, the vulva, the anus, and the likes. And for most people, the infection will be asymptomatic and it will clear on its own. But this is a presentation of someone suffering from papillomavirus, and it presents on the skin. Okay. Then you have double-stranded DNA viruses. So you have the enveloped virus. We've looked at the non-enveloped. This is the enveloped virus. For the family Poxviridae. So Poxviridae, you have the orthopox virus, where you would have the smallpox virus and the cowpox virus, and the monkeypox virus as well. The size, it's bigger, 200 to 350 nanometer. They are large and brick-shaped viruses, as you can see in this picture inserts. All the diseases that are caused by this family of viruses include skin lesions. So that's an important characteristic. And you have the smallpox and the cowpox here. The pox are filled with pus. So these are pus-filled lesions. The enzyme transcriptase is important in their replication. It is the enzyme that initiates the multiplication of the virus. And then the assembly and the synthesis of the virus will occur in the cytoplasm. Then you also have the family Herpesviridae. Important members would be the Simplex virus. They are somehow medium, but quite large, 150 to 200 nanometer. Clinically, they will cause fever, blisters, chickenpox, shingles. So they are the latent viruses. Okay. You would see latencies like shingles, where someone is exposed when they are infants, and it's not going to present again until later on in their life, when it presents again. You have examples of diseases known as infectious mononucleosis, or IM or mono, which is spread through body fluids like saliva, and it can also be spread sexually. I have some more examples and the disease that it causes here in terms of the Herpesviridae. You have the simplex virus, which causes herpes simplex. Varicella virus, which causes chickenpox, shingles. the lymphoid which causes Epstein-Barr virus infections like infectious mononucleosis, which is a form of cancer. We can cause cancer. And you see the symptoms on there as well. So familiarize yourself with this particular slide as well. Then for the single-stranded DNA viruses, so far we've been looking at double-stranded, non-enveloped and enveloped viruses. The single-stranded DNA viruses, we have non-enveloped ones. These are very small in size, 18 to 25 nanometer. That's the Parvoviridae. The important genera is just one, and that's the human parvovirus virus B19. Okay, and that's how it looks like on the micrograph. It causes a disease called the fifth disease. Now, the fifth disease has symptoms like runny nose, fever, headache, but characteristic of it is a rash that is described as the slap cheek rash. It may be associated with painful or swollen joints, and for people who are immunocompromised, it may result in severe anemia. It can easily be spread through respiratory secretions like saliva or sputum from someone who is infected when a person coughs or sneezes, and you are in reaching distance of that person. So this brings us to the end of our lecture on DNA viruses. We'll continue with RNA viruses in the next lecture.